So today we're going to learn about the two most common microscope used in science. One is called the dissecting, and the other one is called the compound. Here we have the compound microscope. The first thing you need to do is to plug it in and turn on the light. We're going to start at the base and work our way up the microscope. The light source is located underneath the diaphragm. The diaphragm also has a little part that allows how much light comes in and out. The light source allows the specimen to become more visible to the human eye. The focus of the slide is controlled by the fine and coarse knobs. The difference between these two knobs is the coarse knobs move the stage up and down, whereas the fine focus knobs just zoom it in and out. There is also a knob on the side of the microscope that allows the slide to move. The stage is the platform for the slide, as the stage clips hold the slide in place. You look into the ocular lenses, or the eyepiece, when wanting to see closely to the specimen. On the opposite side of the body tube is the revolving nose piece, which gives room for the... On the opposing side of the body tube is the revolving nose piece, which gives room for the objectives to move. There are 4 levels of zoom for the objectives. There's the 4 times, the 10 times, the 40 times, and the 100 times. The dissecting and the compound are very similar, but here are some differences. Rather than the whole entire stage, the dissecting has a stage plate on top of the base, which is completed with stage clips. As you can see, there's no light source. You would need to use an external one. In order to control the focus with this microscope, there is only one knob, which is called the focus control. With this microscope, there is only one objective, and in order to mandate the zoom, you use the zoom control knob. The objects you look into are the eye cups, which make it more comfortable to view, and they are attached to the 10 times eyepieces, which are ocular. When you're going to take out a microscope, make sure you do it the correct way. Not like that. Now, the correct way to hold a microscope is to grab it by the neck and the base and to hold it level. We have a bonus microscope for you today, the confocal microscope. Confocal microscopy offers several advantages over conventional wide-field optical microscopy. The ability to control depth of field, elimination of background information from the focal plane, which can lead to image deterioration, the capability to closely examine thick specimens, and the ability to obtain extremely high-quality images from the specimen. Confocal microscopes use spatial filtering techniques to eliminate out-of-focus light and glare in specimens whose thickness exceeds the immediate plane of focus. It's become popular in hospitals because of the growing number of applications it has in cell biology that rely on both fixed and living cells and tissues. Confocal technology is proving to be a major advance in optical microscopy. It's mainly used in dermatology. They'll use a confocal microscope to examine your skin, hair, nails, veins, mucous membranes, and body tissue to see what they need to treat you for. And the best part about confocal microscopes, they use lasers to help you see the image. Pretty cool, right?